This series of videos will cover the structure and function of glands in the intestinal system and complement the earlier material on GI tract by Dr. Al Ghul. Session objectives shown on this slide list the items for studying material. This lecture is divided into two parts. Salivary glands are described in the didactic material and is included in the written test, but you will not be tested on this in the practical exam. As a note, you should review the notes from the foundation block to remind yourself of the structure of glands. Salivary glands are classified as compound tuboacina with lobular organization, where lobules are separated by connective tissue septae. As these are exocrine glands, they are ducts associated with conveying secretion from acini to the surface. When these ducts are present within the lobule, they are called intralobular ducts, and these tend to be smaller in size. As they get bigger, they are seen in the septae and are called the interlobular, or in between the lobule. These terms will be used again when we are describing the exocrine glands in the pancreas. There are three salivary glands with distinct morphology and secretion. Parotid glands only contain serous acini. That is to say, they are producing proteinaceous material. Histologically, we observe ducts that exhibit striation and are involved in modifying the secretions. The gland is often infiltrated with adipose tissue. Submandibular glands are mostly serous but have some mucocini. Striated intralobular ducts are also present. Sublingual glands, on the other hand, have mostly mucous acini with minor contribution by serous. Here, there are no appreciated striated ducts. This slide shows comparative histology of the three salivary glands. The presence of serous and mucous acini can be appreciated in the overview pictures, which are showing darker and lighter stains respectively. Inset on each panel highlights the acinous structure. Note darker and lighter portion of acini in mandibular and the sublingual glands. Darker parts, these appear to partially cover the lighter part. This feature is sometimes referred to as demilume, as highlighted by this marker here and here. Also note the inset in the submandibular panel of striated duct here on the right, showing radial lines in the cuboidal cells. We will discuss a little bit about the composition of the saliva. Saliva is a complex fluid with neutral pH that is composed of mucin, proteins, and high calcium phosphate potassium ions, but low levels of sodium and chloride. As seen earlier, mucins and proteins are produced by specific acini. The striations seen in the ducts are infoldings of mitochondrial basal membrane and function to fashion the saliva composition that is hypotonic. A significant amount of saliva is produced every day, and most of the composition is contributed by the submandibular gland. Functionally, serous secretion provide enzymes that start to pre-digest food, and mucous secretion provide lubrication for chewing and swallowing. In addition to these functions, saliva also has antibacterial and antiviral activities. Secretions from salivary glands are under the control of the nervous system. As histological evaluations, we will not discuss this further. Moving on to the pancreas. This is a compound gland that contains both the endocrine as well as the exocrine portions. The endocrine portion is contained within a well-defined set of cells and is called the Islets of Langerhans. The exocrine portion forms majority of the mass and contains serous glands of tubuloacinar type. The image on the left is an original drawing of pancreas anatomy, showing the main ducts called the duct of Versong. This duct is highlighted in the drawing on the right, together with the pancreas relationship to the gallbladder and the GI tract. This is just for information, so you can appreciate how these structures are associated. Serous acini within the glands secrete lipase, proteases, amylases, nucleotidases, and are stimulated by CCK, which is cholecystokinin, and VIP, vasoactive intestinal peptide. Centroacinar cells are characteristically present at the origin of ducts in the acini and secrete bicarbonate under the stimulation by secretin, a hormone released into the bloodstream by duodenum, especially in response to acidity, to stimulate excretion by liver and pancreas. Acini and centroacinar cells are stimulated by both parasympathetic nervous system and inhibited by the sympathetic nervous system. This is an excellent example of acinar structure, together with centroacinar cells. Note the zymogen granules within the acinar cells that contains the synthetic material for export by intercalated duct. Also note the peripherally located nuclei here and in the picture up here. The centroacinar cells are clearly visible in the middle of the acinus, where the duct system starts. In this picture they are highlighted here, and in the schematic they are highlighted here. The Islet of Langerhans are the endocrine cells of pancreas. The endocrine portion of the pancreas contains a number of cells. The alpha cells produce the glucagon, a hormone that works with other hormones and bodily functions to control glucose levels in the blood. The beta cells produce insulin that allows your body to use sugar. The delta cells produce somatostatin, a growth hormone, inhibitory hormone that affects several areas of the body by hindering the secretion of other hormones. Other cell types within the islets impact gastrointestinal function along with their counterparts in the gut mucosa. This slide shows an overview of pancreas histology. Individual lobules are clearly separated by the connective tissue septae. Within each lobule, a number of islets of Langerhans can be observed as indicated by these black arrows and appearing as lighter in color. Additionally, interlobular ducts can be seen in the lobules as shown by these red circles. Larger ducts outside of lobules are called interlobular and can be seen surrounded by connective tissue in the septae. This is shown in the green circles. There is also appreciable level of blood vessels, but cannot be seen at such a low magnification. You will be able to observe those in the lab. This schematic demonstrates the islet composition as well as spatial distribution of each cell type. Beta cells are primarily responsive to elevated blood glucose levels, secreting insulin to enhance uptake and utilization of glucose by peripheral tissue and the liver. These cells are under the control of parasympathetic by stimulatory signal and sympathetic by inhibitory signal innervations, as well as circulating metabolites and hormones. Alpha and delta cells tend to have a more peripheral distribution in the islet, and they have first access to blood flow to the center of the islets. This arrangement, as seen here in this picture, allows beta cells to be under inhibitory modulation of both somatostatin and glucagon from alpha and delta cells, which are located in the periphery. At another level of control, somatostatin also inhibit glucagon secretion. On this slide, the left-hand panel shows H&E stain, which can distinguish the islets from the surrounding exocrine acini here, but not able to resolve different cell types within the islets. Special stains, as shown on the right side, are required, or immunohistochemistry is required to identify specific cell types. In this case, the alpha cells are being highlighted by this particular silver stain. You will not be held responsible for identifying different cell types within the islets in practical, but you should know their function for the written test.